And thank you so much for the opportunity to talk today. I'm really pleased to get to talk about some of the research I've been doing. So as Annika said, I'm Rachel. I'm a genetics registrar in Wessex, but have been lucky enough to have several years doing research with the Clinical Ethics Law and Society group at Oxford, looking at this kind of question of what's the nature of a genomic result. So the kind of question that I've been having the chance to think about is when we start with a genomic test as our assay, so something that's kind of delineating the 4 to 5 million ways in which our DNA sequence is different to the next person's. What does it sort of mean to make a result from that? What sort of qualities are we looking for in a result, and how do we kind of understand and value a particular genomic variation as being a result? And I think that's particularly interesting in light of the sort of abnormality of any seemingly normal genome. From a typical human genome, we'll all have about 54 variants that have previously been reported as causing disease. Lots of them will look like they wreck the protein that the gene would make, or they're very rare and look like they would change what it does in the body somehow. And because lots of them are very rare or kind of seemingly new enough, it's kind of hard to work out and separate exactly what they might mean for us because there's very little data to go on. So I've been really lucky to have the kind of opportunity to explore how we construct genomic results, so how we sort of arrive at a result from a genomic test by this really interesting methodology called facet methodology. And that's a qualitative research approach where you kind of see your central research problem as almost like a gemstone, and you're trying to look at it in different ways in a kind of strategic way, trying to get flashes of insight on your main research question. So my main question has been, how should we construct genomic results? And I've tried to look at that in different ways, so through how they seem to be represented in stock images about genomic results, how they're written about in UK news items, how they're discussed in this incredible archive of people writing about everyday life in the UK. And I've also spoken to some people experiencing genomic tests and observed some appointments where genomic tests and their results have been discussed. So to start off with, I wanted to talk a little bit about the ways in which popular discourses invite us to imagine genomic results. And to start off, I want to talk about a facet where I looked at how they're kind of evinced in stock imagery. So it's often said that a picture's worth 1,000 words, but I think pictures get relatively little attention relative to text, for example. So often when I'm preparing something, I think quite a lot about the text and what I'd say, and then at the end I'd like bung a picture or two on to sort of glam it up and make it look a bit prettier. But in terms of what you're communicating, I think pictures could often be quite impactful and often perhaps go a bit under the radar because they're kind of a bit harder to pick apart in some ways than text. So I looked through 300 stock images around kind of genomic tests and genomic results. And stock images are sort of pictures that anyone can buy to use on whatever you want to illustrate. So if you're a journalist looking to illustrate a news article, if you want a pretty picture of your leaflet, it's the sort of repository of images where you type in your search terms and it comes up with a load of images that you can buy with particular permissions to reuse. And the most successful stock images tend to kind of end up conforming to existing cliches. So if you think about a story about working from home, they'll have like someone on a sofa with a laptop, or a story about freedom will have someone kind of jumping in the air like that. Because for a stock image to be successful, it does need to feel appropriate to the context you want to use it in. But there have to be a lot of contexts where people would feel that about it in order to get bought by a lot of people. So these are some of the ones that were coming up as the most relevant when you type genomic test or genomic result into popular stock image banks. And 31% of images featured double helix. And this is a sort of illustrative recreation I made with Canva AI, but I think this is quite representative of how it was often displayed. So you'd have it, the main focus of the image, set apart on this sort of dark celestial type background and kind of glowing with light and looking, potentially quite impressive. Gel electrophoresis derived patterns, like I'm showing in the middle one, were kind of shown in 16% of the images. And again, I think the way they were delineated is they often looked kind of a bit barcode-like. There'd be bands that were glowing sometimes they'd be partnered with people kind of reacting around them, like holding a gel negative up to the light or pointing at a particular glowing band. And I think, again, this genetic material or data were often foregrounded in these images, potentially in a way that showed sort of illumination being read fairly directly from the data. Scientific equipment was often foregrounded as well. So 15% of images featured a pipette, and often that was the kind of focal point of the image, usually like the droplet dangling on the end of it, potentially inviting ideas of kind of accuracy, precision, immediacy. And actually microscopes did better than pipettes. They were there in about 20% of the images, despite probably not being that prominently part of most genomic testing pathways. 54% of the images had no people in them at all. So kind of data and equipment tended to do better. And for example, that image on the right, I'll have counted that if this were one of the ones in the thing as including a person, because there is a bit of a glove in the corner of it. But often that kind of, I think, shows how many of the images, even if there were people, it was the equipment that took centre stage. So you'd have like a scientist staring down a microscope, or a sort of person in a white coat, like pointing at a double helix or something. It kind of, to me, I think a lot of the stock images gave a message of insights or illumination, zooming pretty directly at the code, and people sort of serving that process by pipetting the stuff that was needed, or by reading off the annotation on the double helix, but less kind of showing people having a prominent role in either sort of participating in testing. It was 5 of the 300 images showed someone in a kind of the role of having a genomic test, or in kind of constructing meaning from it. And it sort of reminded me of this quite old quotation of how DNA has assumed a cultural meaning similar to that of the biblical soul. And I think kind of having double helixes showering sparkles in a sort of spacey type background does arguably tap into that a bit. And these images do sort of get everywhere. So you might recognise this as the front cover of the recent NHS 10-year health plan for England. And I think, again, it kind of makes the point probably about how images might be chosen as an afterthought, probably. So probably they thought quite a lot about what to write in it, and then a design consultancy or something made it look nice. I don't know if that's how it works, but like, what's the kind of message that might give to somebody looking at it? It's probably that being fit for the future, DNA might be important, and it might have impressive things to do. And moving on a bit from those to stock images in context, I went on to look at some UK news items discussing genomic tests and their results. But I think, in fairness, for people who didn't actually read the articles I'm going to go on to tell you about, probably the headline and a stock image will have been all they saw. So if they didn't read that article, they'd have got the kind of message about DNA testing of newborn babies with a picture of a cute baby with a double helix with annotations hovering over their cot to give the kind of message of the story. This one about a gene testing revolution, which, in the middle of it might have had all sorts of nuances about, how revolutionary is it now? How revolutionary will it be in X years' time? But if you've got a big picture of a kind of DNA code and a magnifying glass and it says cancer pretty clearly written in it, that in itself gives a message as to what we might expect from genomic testing. So I undertook a kind of framing analysis of 186 UK news items that discussed various genomic initiatives. So DDD, the 100,000 Genomes Project, the NHS Genomic Medicine Service, Generation Study in Our Future Health. And this was in kind of various different prominent UK news items. And a lot of them would sort of start by giving the context of this sort of drumroll introduction to genomic testing. So they'd contrast the past, where it took 13 years of painstaking research to read the first ever complete genetic blueprint for a human, compare that scientific breakthrough to splitting the atom and putting a man on the moon, and then say kind of today, nearly 15 years on, this was in 2017 they're writing this, cutting edge technology means sequencing the 3 billion letters of DNA that make up a human genome takes just 3 days. So quite often stories would be set up such that you'd get the impression that it was sort of sequencing capability and cost and time that was holding us back. And now we can do all that really quickly, nothing can stop it. In fairness, I think a lot of the articles did allude to the complexity of the task and the scale of the data, and the challenge of that in constructing a genomic result, but it was usually as context to a story where genomic testing had produced a helpful result. So in the kind of the stories I read, there were, I think, 85 specific instances where it discussed a diagnostic genomic test of a specific person living with symptoms suggestive of a rare disease, and 88% of those described having a diagnosis. Quite a high proportion of those were treatable, and that's a bit at odds with the sort of 40% figure that you probably give in clinic. In the 12% there, several of them were stories that had all featured in just one article about life with an undiagnosed condition. So if you'd happened to miss that one, the proportion would have been even more favourable towards the likelihood of making a diagnosis. And a few of the others were ones who'd been interviewed sort of like at the moment of joining a project, so you wouldn't necessarily expect them to have a result kind of seconds later, and you might be left with the expectation of these, helpful results to come. In the context of screening, I think often articles didn't particularly differentiate, especially in the context of newborn screening, between diagnosing and making a prediction from the genomic code. And there was often a bit of a false equivalence where an article would contrast the sort of future opportunity of something like newborn screening with the past of long diagnostic odysseys. And it would sort of present it as if the choice was newborn genome screening now or waiting for years and years, not knowing why your child was mysteriously ill, and the option of investing heavily in kind of rapid diagnostic testing for people once they start to develop symptoms didn't get much airtime. And obviously this is a bit of a kind of caricature of what the article said, and there were some which had dissenting views, but quite often they played into this picture of genomics as almost this panacea, which would shift from situations of ill health to a society where health problems were prevented. That would be cheaper for everyone to prevent rather than cure. And this is a quotation from an article that Keir Starmer wrote while he was leader of the opposition about, with the power of genomics, we could help every one of us live a healthier life, prevent more illness, and take more care of our lifestyle choices. So to summarise, I think the stock imagery in UK news items that I looked at kind of presented this idea of genomics as almost like hidden gems, where genomic results would be very valuable once found, quite fixed in nature and qualitatively different to the genomics variation that surrounds them. So it might be quite difficult to find a result, and you might need very complex technology to do it, but once you spot it, you'll know what it is. So to move on to a sort of somewhat related question of what features do people conceptualise genomic results as having, I was lucky to kind of explore this through looking at what people wrote in response to the Mass Observation Project. And this is this incredible initiative that's been running for many years, but invites people across the UK to write in response to various directives 3 times a year. And in spring 2022, Cells did one, which was led by Susie, where we sent out a directive on genetics and health in our everyday lives, which went out along with another directive on the royal family and one on grudging acts to the kind of bank of observers, and 147 responded to our directive. And one of the questions we asked was, what comes to mind when you think about genetics? And people were mentioning, school biology lessons, Mendel's peas. I was quite pleased that a lot of people referred to images of long molecules and codes and models of double helix and laboratory scenes, because that kind of tied in with what I'd seen in the stock images. But people's ideas were being influenced by many sources over many years. A lot of people who had personal experience of genetic testing or knew someone who had, but often it had been to explore ancestry rather than kind of NHS health genetic testing. And responses encompassed areas like forensic testing, where kind of teeny amounts of DNA were seen as really proving the difference between kind of guilt and innocence, proving relatedness, where this was seen as really powerful information with potential to sort of uncover family secrets and cause disruption. Ancestry tests, where observers were often quite kind of scathing about the idea of quantifying ancestry, but and archaeological genetics, where in the a lot of people, sorry, not a lot, but a few people referred to the story of where they identified the skeleton of Richard III and how conclusive they'd found the DNA evidence as being in that. So people were bringing in a lot of different exposures into their kind of thoughts about genetics and health in our everyday lives, and didn't make a kind of a clear formal distinction between sort of this was the genetics of 1990, this is the genomics of now. It was a sort of a continuum that people were drawing on and bringing into their answers. And I think there was often a sort of implicit expectation of clarity and power from genomic results. So these are a few quotations from observers reflecting on the Newborn Genomes Programme. So the idea of knowledge being power, the value of being forewarned, or kind of while it could be devastating to discover, there could be the potential for genetic disease, ultimately only be a good thing to learn this as soon as possible. Or perhaps another one which I guess would see this information as poisonous, but nonetheless kind of clear and powerful. Somebody saying they didn't think they'd want to know the death day of their newborn baby. But I think along with that expectation of clarity and power, observers were also challenging that idea at lots of points throughout their answers. And I think this quotation, which I think is my favourite from the data set, from a librarian in his 60s, I thought just beautifully captured quite a lot of what it was seeing. He said there's something quasi-magical, alchemical might be a better word, about the instinctive popular attitude to DNA testing and the research into its practical uses. It's hard not to think of it in very literal terms, that pulling a gene from one position to another or excising it can immediately change the physical appearance or personality traits or directly influence the course of a disease or chronic medical condition. And he mentioned that I'm sure that's all fantasy, but it's a creepy feeling nonetheless to feel oneself in all one's human complexity reduced to whatever a genome sequence looks like. I'm imagining it as a Manhattan Yellow Pages thick tome filled with variations on ATCG. And I thought that kind of showed how, while people were sometimes kind of this hidden gem or like default concept of genomic results as having clarity and power was really hard to get away from, people were also resisting it in other areas of their answers and showing far broader ideas around genomics. So then I had a bit of a look at what features genomic results appear to have in clinical settings, and these come from observing a few clinical appointments where genomic tests and their results were discussed. And in that context, it seemed much clearer that genomic variations, rather than sort of shining or beeping out the code, they're just one piece of the puzzle, and it took more to create a result. So the most sort of hidden gem type result I probably observed being discussed was one which actually at the start of the appointment had been seen as a variant of uncertain significance. And in meeting the child and seeing that she very closely matched the kind of symptoms that children tend to have who've got variations in that particular gene, that was what kind of transformed it to a result in that moment. So the clinician mentioned it's like a jigsaw puzzle, does everything fit together well enough? And in various other appointments, I saw things where kind of the puzzle pieces were maybe being kind of drawn together, but it wasn't quite clear that it did fit together well enough, or things were changing. So they said we need the muscle biopsy and dad sample to add it all together in one example. And often outcomes from genomic tests, rather than being really powerful and clear, they seemed sort of drifting around, and there was a provisionality in the way that people were engaging around them. And clinician did allude to that. So I realise it can be really frustrating for families that we seem so cautious in making a firm diagnosis, but we want to be sure it's right. And sometimes you've got the sense that kind of if this child came back in 5 years' time and scooped the data again, would it be the same variant that was drawing people's attention, or would it be something totally different? Would it have just drifted away? It was also quite clear that results weren't static and weren't just sort of made and fixed in a moment of an appointment. They would go on to be drawn on beyond the clinic in different ways and for different purposes. So one mother in the middle of an appointment, we're discussing some genomic information, said, I just want to make sure I've got it so I can tell other people because I'll have lots of questions. I'm not a massive Googler, but my family are. So you could just see this kind of result being discussed in an appointment that would then go on to be communicated to family, who might then do their own kind of looking into it, or might be in contact with patient groups, and school might have an interest in knowing it, and all these sort of factors around the family that could be drawing in on a result and using it in different ways in different contexts. So I don't love this analogy. I'm really struggling to come up with a better one than a hidden gem, but I think it almost seemed more like surveying an ecosystem where you've got kind of a noisy and varied landscape of genomic variation, and different things might be important at different times. things might shrivel away or grow to kind of take over the patch. And the challenge is determining what's important and when and why. So here, is it those like dandelions that might give them a big weed problem? Is it this weird little red flower that seems to be on its own? As in, what do you pick to be the sort of important thing to communicate from all of that noise? And just to discuss kind of some interviews I did with people experiencing genomic tests. I think when we were discussing results in the abstract, it often, our kind of stock concept between both me and the people I spoke to did seem to be sort of this hidden gem idea. So one interview I was speaking to a woman who was taking part in a trio trying to work out why her child had a particular health problem, and she said to me they'd found that the child had the same mutation, she said, as about 5 other people. There hadn't been much from it. But when we were exploring why it would be so important to have a genomic explanation, she was saying it just clinches it somehow, makes it solid. Right, we know what it is, we can now do something about it. And then on another occasion, I was talking to a woman who'd been having trio testing to try and explain quite a lot of unusual medical things that her child had experienced. And we were talking about kind of grey areas or kind of uncertainties in genetics. And she said, it is what it is, it's black or it's white. It tells you what it is, doesn't it? So how can that be a grey area? Yet another point, she was very much sort of discussing uncertain information and whether and when she'd value it. So I think some people did have experiences that sort of chimed with the hidden gem model. This was one quotation from somebody I spoke to whose child had a very helpful diagnosis through the project, and said, I'm amazed because I have visions of these thousands of genes being sifted through, and to think that they found one. But then there were kind of other experiences too. So this was from somebody I spoke to trying to find a reason why they had particular muscular problems, and the project hadn't found clear answers for him at that point. And he said, I was a bit disappointed, but I can understand why. It's trying to find a needle in about 10,000 haystacks. And then other people were living with quite challenging or very uncertain information as a result of genomic testing. This is a quotation from a family I spoke to who'd had a genomic test trying to explain why their young daughter was deaf, and had found something uncertain that, if correct, would confirm why she was deaf, but would also predict future sight loss. And they had not been aware that was a potential outcome from the test, and the child was too young to have any kind of testing that could give clarity on that. And they described it being a really sort of unsettling and unanticipated outcome from their test, and it had really made them reconsider what they would feel would be valuable as a result, because they said at the outset, they were like, tell us everything, we want to know everything. And having been through this experience, they were like, actually, we want to know what we can do about it, and we'd be a lot more kind of, it's a lot of a harder decision in retrospect now, living with this outcome. Here's another quotation from a person I spoke to whose child was having trio testing trying to explain her child's developmental delay, who felt quite cheated by her experience of genomic testing, and I think possibly because of the sort of power and prevalence of a hidden gem model and the way we talk about genetic tests as reading the genetic code. She just couldn't understand why she hadn't got a clear diagnosis from a test that had ostensibly read her whole genome. She said, I didn't mind it was going to take a while, I was going to look for every possible spelling mistake. I felt like we couldn't do any better, when in reality that wasn't true. We kind of felt that someone would go in with a metal detector and look for them. So I think the kind of interviews and clinic experience sort of gave me the impression that these hidden gem type results that get so much airtime in popular discourse are sort of the tip of an iceberg, and they get lots of attention from kind of in political discourse and in kind of news stories and in stock imagery, but there's a lot below the surface that doesn't get that same airtime. And yet these are potentially people's default concept when thinking about genomic tests, because that's the way it's sort of thought and talked about and represented in the popular discourse. And a question I asked a number of people about their genomic test was kind of, when did you decide you wanted it? Was it a difficult decision? And many people were saying things like, as soon as it was offered to us, we knew we wanted it, or we were set on doing it. There's never a question that we weren't going to do it. And I think that kind of comes with the risk that consent conversations don't prepare people So the woman who I mentioned who felt quite cheated by her experience of genomic testing, when I was talking about the consent process with her, she kind of mentioned, I thought it was ridiculous. I feel the NHS already will have had robust ethical procedures. I could trust in it without it being explained in an hour and a half appointment. I think it was excessive. Yet evidently it hadn't fulfilled the purpose it was hoped to, in that she then had felt quite kind of, her outcome had not matched what she had felt was what she would be expecting from the test. She felt there should be more transparency about the panel-based way data might be looked at. And so I guess reflections on that, I think these hidden gem type experiences of genomic results, it's because it's like very lovely and natural to celebrate the successes of genomic testing, but they do then get lots of airtime in popular discourses around genomic testing, to the point where if people don't work with genomic data on a regular basis, as I guess most people don't, that and being taught about it in a very Mendelian kind of binary way at school possibly gives the expectation that results like this come as standards, and other experiences are given so little attention that then that kind of adds to the isolation potentially of people who experience those in the context of rare disease diagnosis. And it also sets up quite unhelpful conversations when it comes from moving diagnosis to screening, because the sort of uncertainties that might arise from rare disease screening and the sort of limited predictive power of common disease screening, where other environmental factors might matter a lot more than a kind of genetic complement, are sort of minimized. And the discussion then gets into, should we open this Pandora's box of the genome and let these hidden gems come out, rather than whether and when and why we should value particular variations in our genomes as being results. So returning to the sort of central question, I think, about how we should construct genomic results, I think there's a case for having wider concepts of results that try to encompass complexity and subtlety and contingency, and sort of recognizing that these powerful and transformative results that get a lot of attention in news items, that are sort of implicitly shared in many stock images, are sort of the tip of a much bigger iceberg, even in the context of rare disease diagnosis, which is sort of the quintessential setting where you'd expect genomic testing to kind of come up with the goods. In a screening context, a hidden gem concept of genomic results is perhaps likely to be even less helpful. In terms of things that could possibly be useful, I thought trying to be aware that a sort of hidden gem type concept of genomic results is quite powerful, quite prevalent, and might be understandably people's starting point when coming to a conversation about genomic testing. So potentially sort of recognizing and challenging that a bit explicitly to allow consent conversations to do the job you hope, rather than sort of be something that people sit through in order to get this sort of transformational test that they know already that they want. Finding ways to sort of amplify a range of outcomes and experiences for genomic testing, rather than only the very best experiences, and potentially kind of exploring language that's used around genomics, like reading the genetic code and thinking about images that are used, like sparkly double helixes. So thank you so much for listening.